Hello everyone, welcome to Concept and Fact of Life Sciences. Today in this video we are going to discuss about the nuclear genetic material that is known as the chromatin. Now the chromatin, it is made up of two words which are chrome and the matin. Chrome means the colorful while the matin means the thread. So these are the colorful threads. Why these are called as colorful threads? Because if you stain the nuclear genetic material with some nuclear stainers like acetocarbon and observe them under the microscope, they will appear in the form of colorful thread. वो आपको रंगीन धागे नुमा देखने को मिलेंगे. That's why it is called as chromatin or the colorful threads. Now the next question is, what is the need of packaging? What is the need to carry out the conversion of this chromatin into the chromosomes? Or we can say what is the need to carry out the condensation of DNA. समझने की कोशिश करते हैं इसको. If we talk about the size of nucleus in case of eukaryote, it is around 5 to 10 micrometer, or we can say 10 to the power minus 6 meter, or we can say that it is 1 millionth of the meter. And at the same time, if we talk about the size of genome, in case of haploid cell, it is 3.3 into 10 to the power 9 base pair, which is equivalent to the 1 meter. While in diploid cell, it is 6.6 into 10 to the power 9 base pair, which is equivalent to 2 meter. Now we have to arrange this much size of genome within this much size of nucleus. For this, we have to carry out the condensation, or we have to organize the genome. So for this, there is a genome organization. Now we have different levels of genome organizations, which includes, here we will discuss about different levels of genome organization from chromatin to the chromosomes. So if we talk about the chromatin, these are the 2NM fibers, which solely includes the DNA. So it is also known as the 2NM fiber. Now this 2NM fiber, they further associates with some proteins which are called as histone proteins. There are 4 different types of histone in the form of H2A, H2B, H3 and H4. They will form a structure which is called as the nucleosome core. Now this chromatin, it interacts with this nucleosome core, which is made up of histone protein, and forms the structure which is called as nucleosome. जिसे अपन कहते हैं nucleosome. If you observe the nucleus under the electron microscope, under this nucleosome stage, it will appears in the form of sting on beads. वहां पे आपको एक मोतियों की माला जैसी संरचना देखने को मिलेगी. That's why it is known as sting on bead model. जहां पे sting जो आपके धागा है वो होती है DNA, और जो beads हैं these are the nucleosome core made up of histone. So in this nucleosome we have two structure, one which is known as the DNA, while the other is known as the nucleosome core, which is made up of histone protein. Okay? So this is the second level organization which is known as nucleosome. Further, we have another histone which is known as a linker histone. Linker histone, and it is the H1 histone. Now this H1 histone, it is not the part of nucleosomal core. We can say after the formation of structure which is known as nucleosome, then there is a binding of this H1 linker histone. Now this H1 linker histone, it generally attaches between the two nucleosome. दो nucleosome के बीच में attach करता है, that lead to the conversion of this nucleosome into the next level of organization which is known as chromatosome. Now we have the nucleosome. There is a presence of nucleosome, and this nucleosome, it includes chromatin plus nucleosomal core, and there is a binding of H1 linker histone. That's why this structure is now known as the chromatosome. इसलिए अपने पास में क्या बन गई स्ट्रक्चर, अब chromatosome की बन गई. The binding of this H1 linker histone to the nucleosome will convert this nucleosome into the level 4 organization that is known as a 30 nm fiber. Now, for this 30 nm fiber, we have two different hypotheses. One is known as solenoid model, while the other model is known as zigzag model. That means इस 30 nm फाइबर की जो साइज है वो तो 30 nm ही रहती है, बट ये स्ट्रक्चर कैसा है उसके लिए दो हाइपोथेसिस हैं अपने पास, एक सोलेनॉइड मॉडल है, एक जिगजैग मॉडल है। दोनों ही जो मॉडल है वो वेल एक्सेप्टेबल है। ठीक है, तो बात करेंगे इनके बारे में आगे अपन। यहां सिर्फ इतना सा समझना आपने को, फर्स्ट वी हैड a 2 nm fiber that is of the chromatin. It interact with the histone proteins, or we can say with the nucleosomal core, that lead to the formation of a structure which is known as nucleosome, which is a 10 nm fiber. Now, after binding of H1 linker histone, the nucleosome it will convert into the structure which is known as 30 nm fiber. Now, in this 30 nm fiber, if we talk about the solenoid model, दूसरे मॉडल के बारे में आगे अपन डिस्कस करेंगे। If we talk about the solenoid model, then the 6 nucleosome they will arrange in the form of a helix. Okay, वो कुछ इस तरीके से, अगर 3D स्ट्रक्चर की बात करें, 5, 6, these 6 histone they will arrange in the form of a helix and will lead to the formation of a structure which is hollow from the center. That's why it is known as the solenoid. और इसकी जो साइज होती है that is 30 nm, इसलिए इसको अपन कहते हैं it is a 30 nm fiber. So binding of H1 linker histone will carry out the further condensation, lead to the formation of structure which is known as 30 nm fiber. Now this 30 nm fiber it further interacts with some non-histone protein, है ना? Now there is an interaction with non-histone प्रोटीन। अभी तक जो इंटरेक्शन था डीएनए का वो किसके साथ था? हिस्टोन प्रोटीन्स के साथ था, चाहे वो H2A, H2B, H3, H4 हो जो बना रहे थे न्यूक्लियोसोमल कोर, और चाहे वो लिंकर हिस्टोन हो जिसे अपन कह रहे थे H1. नाउ देयर इज अ एसोसिएशन ऑफ दिस 30 nm फाइबर विद सम non-histone proteins, and these proteins they are called as scaffold proteins, which includes some SMCs, that is structural maintenance of chromosomes protein, that are condensins and the coesins जो further इसका condensation करेंगे. So there is an association of 30 nm fiber with some non-histone proteins which are known as scaffold proteins, and this interaction it occurs through, we can say, the SAR regions of the 30 nm fiber. The SAR regions, that is scaffold associated regions of this 30 nm fiber, they interacts with the scaffold protein. These are the AT rich regions that interacts with these scaffold protein. Now the interaction of this scaffold protein with the 30 nm fiber will carry out the further looping. Now we are having a, suppose it is a solenoid, and these solenoid now they will coil in the form of loops on these scaffold protein, or with the help of these scaffold protein. These scaffold proteins they helps in the formation of these loops. For example, if we have the condensin, these condensins they helps in the formation of loops, and due to the formation of loops, now the structure we will observe it is known as a 300 nm fiber. अब जो 30 nm फाइबर है, आफ्टर एसोसिएशन विद सम नॉन हिस्टोन प्रोटीन इट विल नाउ बिकम 300 nm. देन फर्दर द कंडेंसेशन इट कंटिन्यूज, जो कंडेंसेशन है वो कंटिन्यू रहता है. देयर इज अ फर्दर कंडेंसेशन एंड इट विल लीड टू द फॉर्मेशन ऑफ ए structure which is now having the size of 700 nm, and it is known as the chromatid. Now the chromatid they have a centromere. Now the two chromatids which is having the size, each having the size of 700 nm, they interact with each other through their centromere with the help of some other SMCs, that is structural maintenance of chromosome protein, which are known as coesins, जिनके बारे में आगे बात करेंगे अपन. Through the coesins they interact with each other, lead to the formation of a 1400 nm fiber that is known as the chromosome. So in this way we can say there is a conversion of this 2 nm fiber into the 1400 nm fiber, which is known as chromosome, or we can say there is a conversion of chromatin into the chromosome. Now क्यों ये कंडेंसेशन हुआ, एक चीज तो अपन ने पढ़ी, वी हैव टू पैकेज द न्यूक्लियर जेनेटिक मटेरियल विद इन द न्यूक्लियस. साइज बहुत बड़ा है जीनोम का, न्यूक्लियस का साइज बहुत छोटा है, इसलिए उसको पैक करना है उस साइज के अंदर, तो आपको कंडेंस करना पड़ेगा. सेकेंडली, दिस इज प्री रिक्विसाइट फॉर द सेल डिविजन ऑल्सो. अगर आपको सेल को डिवाइड करना है, न्यूक्लियर जेनेटिक मटेरियल को इक्वली डिवाइड करना है, इन दैट केस यू आल्सो रिक्वायर टू कंडेंस द डीएनए. सारा जो सामान है, जो भी सेल का मटेरियल है, उसको आपको एक जगह पे लाना पड़ेगा टू डिवाइड देम इक्वली. सो फॉर द सेल डिवीजन दिस कंडेंसेशन इज आल्सो रिक्वायर्ड. ठीक है. नेक्स्ट अगर अपन बात करें एक एक करके इन सब स्ट्रक्चर्स के बारे में, स्टार्ट करते हैं अपन न्यूक्लियोसोम के साथ में. सो इफ यू टॉक अबाउट द न्यूक्लियोसोम under the electron microscope, जैसा कि अपन ने बात किया, if you observe the nucleus under the electron microscope at the nucleosome stage, then it will observe in the form of a sting on the bead model. The DNA, it remain coil around the protein which forms the structure which is called as nucleosome, जो आपको ऐसा कुछ observe होगा जैसा अपन ने डिस्कस किया, and it is known as the 10 nm fiber. Now in this nucleosome we have two structures or two components, we can say. We are having the DNA as well as the proteins. Now the DNA, as we all know, that it is made up of three things. We are having the nitrogenous base in the form of A, T, G and C. We are having a pentose sugar and a phosphoric acid. आपने क्या लिखा? DNA is acidic in nature. अगर ये acidic nature का है तो इसके ऊपर चार्ज कैसा हो जाएगा? It is having the negative charge. The DNA, they contains the nitrogenous bases, but still they are acidic in nature. Why? Because the acidity which is provided by the phosphoric acid, it is far higher than the basicity which is provided by these nitrogenous bases. That's why the DNA is acidic in nature, having the negative charge. Second component of this nucleosome, it is the histone. Now the histone, these are the basic protein. As we know that histone proteins, they are the polymer of amino acid, और जो amino acid histone को बनाते हैं, these are basic amino acid which are lysine and the arginine. That's why these histone, they have the positive charge. और अपन ने स्टार्टिंग के अंदर जब केमिकल बॉन्डिंग पढ़ी थी, उसमें अपन ने कहा था द डीएनए इट इंटरेक्ट्स विद द हिस्टोन थ्रू द इंटरेक्शन व्हिच आर नोन एज ionic interaction. These are not the ionic bonds, these are the ionic interaction which occurs between the molecules which have partial negative and the partial positive charge. So the DNA, it can interact with the histone with the help of certain ionic interactions. Now we have 5 major types of histone in the form of H1, H2A, H2B, H3 and H4. Now each of these 4 histone, or we can say 2 units of each of these 4 histone, that is H2A, H2B, H3 and H4, they together forms the structure which is called as nucleosomal core. जैसा कि अपन ने कहा था, nucleosomal core किसकी बनी है? H2A, H2B, H3 और H4. इनकी कितनी यूनिट्स होती हैं? 2. So we can say this nucleosomal core, it is a octamer. 8 सब यूनिट्स हो गई इसकी. 2 H2A की यूनिट है, 2 H2B, 2 H3 और 2 H4 की. तो टोटल नंबर ऑफ यूनिट्स 8. So it is a octamer which is having 2 units of each 4 histone, जो 4 histone ये है. Now this H1, it is not the part of nucleosomal core. अपन ने कहा था it is a linker histone. बात करते हैं how the assembly of these histone it occurs. So there is a histone assembly. First of all, we are having the histone in the form of H3, H4, we can say individual units. First, there is a formation of a heterodimer through the interaction of H3 and H4. So H3, H4, they interact with each other, leads to a formation of a heterodimer. Then there is a formation of heterodimers of H2A and H2B. H2A और H2B भी आपस में interact करते हैं और क्या बना देते हैं? Dimers बना देते हैं. These are called as heterodimers because they are having 2 different type of histones. Then these heterodimers, they further interact that lead to the formation of a structure that is called as heterotetramer, जो क्या बना देते हैं, एक हेटेरोटेट्रामर का फॉर्मेशन कर देते हैं। ये H3 के साथ में इंटरेक्ट करेगा और एक 4 सब यूनिट कॉम्प्लेक्स बन जाएगा जिसे अपन कहेंगे हेटेरोटेट्रामर। देन these heterodimer, they interacts with this heterotetramer in the form of dimer, means यहाँ पे heterotetramer नहीं बनता है, जो डायमर है H2A H2B का, it directly interacts with these heterotetramers, so that leads to the formation of a structure which is called as octamer, and it is the nucleosomal core we can say। ठीक है, तो क्या समझने की बात है यहाँ पे कि वी आर हैविंग a heterotetramer of H3 and H4, and the interaction of this heterodimer, it occurs in the form of dimer। यहाँ पे ये टेट्रामर नहीं बनाएंगे, ये डायरेक्टली इंटरेक्ट करेंगे इस ऑक्टामर के साथ में इन द फॉर्म ऑफ ए डायमर, और जैसे ही ये इंटरेक्ट करेंगे इट विल लीड द फॉर्मेशन ऑफ ए स्ट्रक्चर विच इज नोन एज ए ऑक्टामर, एंड इट लीड टू द फॉर्मेशन ऑफ न्यूक्लियोसोमल कोर, जिसे अपन न्यूक्लियोसोमल कोर कहते हैं। Now, if we talk about the conservation of these histone, the most conserved type of histone, it is the H3. Conserved means the histone they are made up of amino acids, और अगर आप H3 को कंपेयर करेंगे ऑफ डिफरेंट ऑर्गेनिज्म तो आप देखेंगे ड्यूरिंग द कोर्स ऑफ इवोल्यूशन उसकी जो अमाइनो एसिड सीक्वेंसेस है उसके अंदर सबसे कम बदलाव देखने को मिले हैं। दैट्स व्हाई वी कैन से दैट द H3 इट इज द मोस्ट कंजर्व टाइप ऑफ हिस्टोन। और सबसे ज्यादा वेरिएबल, सबसे ज्यादा वेरिएशन कहां देखने को मिली है आपको? इट इज ऑब्जर्व्ड विद इन द H1 टाइप ऑफ हिस्टोन। So in this way, there is a formation of a nucleosomal core. Now, the nucleosome, it is made up of two things. One is the nucleosomal core, which is made up of histone, and other thing is the DNA. So we are having the nucleosomal core, 2 units of each 4 histone that lead to the formation of a octamer, which interacts with the DNA. Now, the DNA, which is the part of one nucleosome, it is 146 plus 2260 base pair of linker DNA. So we can say the DNA which is a part of one nucleosome, it is around 200 base pair. ये याद रखना है आपको, it is around 200 base pair। दो पार्ट्स में डिवाइड किया है, एक आप कह रहे हैं 146 बेस पेयर और दूसरा 2260 बेस पेयर, दैट इज द लिंकर डीएनए। जिस तरीके से लिंकर हिस्टोन है, वी आर आल्सो हैविंग द लिंकर डीएनए। नाउ दिस 146 बेस पेयर ऑफ डीएनए is the DNA that directly interacts with the histone। ये जो DNA आप देख रहे हैं जो डायरेक्टली इंटरेक्ट कर रहा है हिस्टोन से, इसका जो साइज है इट इज 146 बेस पेयर, while this part which is linking the two nucleosome, दो न्यूक्लियोसोम को जो लिंक कर रहा है इसको अपन कहते हैं लिंकर डीएनए, एंड द साइज ऑफ दिस लिंकर डीएनए इट वेरीज फ्रॉम 20 to 60 बेस पेयर। ये वेरिएबल होता है, कुछ ऑर्गेनिज्म में 20 बेस पेयर देखने को मिलते हैं, कुछ में 60 देखने को मिलता है, बट अपन कह सकते हैं कि जो रफली एक आइडिया ले सकते हैं कि जो न्यूक्लियोसोम का पार्ट होता है डीएनए दैट इज अराउंड 200 बेस पेयर। ठीक है, तो दूसरी चीज यहां जो समझने वाली है वो है we are having 1.67 turns of DNA around one nucleosome, यानी कि एक कंप्लीट राउंड इंटरेक्ट करता है, देन एक 3/4 राउंड कंप्लीट करने के बाद में वो लीव कर देता है फॉर द नेक्स्ट न्यूक्लियोसोम। लाइक यहां समझने की कोशिश करते हैं इसको, वी आर हैविंग वन कंप्लीट टर्न, उसके बाद में एक 3/4 टर्न पूरा हुआ, देन इट विल लीव द nucleosome and interact with another nucleosome. So we can say that in one nucleosome the DNA it completes 1.67 turn, or in other words we can say it completes 1/3/4 टर्न्स। ठीक है, एक पूरा और एक 3/4 टर्न पूरा करने के बाद में एक न्यूक्लियोसोम को वो लीव कर देगा। ठीक है, सो क्या चीज यहां से क्या आउटपुट निकलता है कि द डीएनए व्हिच रिमेन इंटरेक्टेड विद द हिस्टोन इट इज 146 बेस पेयर, द लिंकर डीएनए साइज इट इज 2260 base pair which links the two nucleosome, and in one nucleosome the DNA covers or DNA completes 1.67 turns। ठीक है, now we have next phase, next level of organization that is known as the chromatosome। In chromatosome there is a binding of another histone which is called as the linker histone, and this linker histone, it is known as H1 histone, जिसे अपन कहते हैं H1 histone. Now the molecular chaperone which facilitates the binding of this H1 linker histone to the linker DNA, it is known as NAP1. That is simply the nucleosomal, we can say assembly protein 1, nucleosome assembly protein 1, NAP1. It is a molecular chaperone which facilitate the binding of this H1 linker histone onto this linker DNA, or we can say onto the nucleosome and the linker DNA. कैसे बात करते हैं, बाइंडिंग कैसे होती है उसके बारे में बात करते हैं. So we are having the H1 linker histone which interacts with the nucleosome as well as with the linker DNA, जो न्यूक्लियोसोम के साथ भी इंटरेक्ट करता है, और वी कैन से, वापस ले लेते हैं इसको, it will bind over here, will interact with both the nucleosome as well as with the linker DNA. जो linker DNA दो nucleosomes को लिंक करा था, that size around 20 to 60 base pair depending upon the organism. So it interacts with both nucleosome as well as with the linker DNA. It is having two tail. One is known as the N tail, while the other tail is known as the C tail. It is having N and the C tail. Now it further covers 20 base pair of the linker DNA. We can say 20 base pair का लिंकर डीएनए ये कवर कर लेता है. जो आपका मान लीजिए अपन 60 बेस पेयर लेके चलते हैं, उस 60 बेस पेयर में से 20 बेस पेयर it will be covered by the binding of this H1 linker histone. On this basis we have a concept of nuclease-protected and the nuclease-susceptible DNA. So if we talk about the DNA which is nuclease-protected, the DNA which remain interacted to the histone, that is this 146 base pair of DNA, it is nuclease protected. कोई भी न्यूक्लियस, जो न्यूक्लियस वो एंजाइम्स होते हैं जो न्यूक्लिक एसिड का डिग्रेडेशन करते हैं, तो जो न्यूक्लियस है वो इस 146 बेस पेयर ऑफ डीएनए को डिग्रेड नहीं कर पाएंगे. सो वी कैन से दैट इट इज अ न्यूक्लियस प्रोटेक्टेड डीएनए, व्हाइल this linker DNA, it is the nuclease susceptible DNA. ये जो linker DNA है ये nuclease susceptible है, which is around we can say 60 base pair. But after the binding of H1 linker histone, it further protects 20 base pair of DNA. Means पहले 146 तो था ही, after binding of H1 linker histone, अब protected जो DNA है उसका साइज कितना हो जाएगा, 166 base pair हो जाएगा. So we are having 166 base pair of DNA which is nuclease protected after the binding of H1 linker histone, और जो nuclease susceptible before the binding of H1 linker histone जो था 60 base pair, वो अब कितना रह जाएगा, it will now become 40 base pair. So we can say there is a concept of nuclease susceptibility and susceptible and nuclease protected DNA. ठीक है. Now the next concept is that the entry and the exit of DNA within this nucleosome, it is through the H3 and H4. We can say the entry and the exit of DNA from the nucleus, it is through the H3 and H4 histones. The H2A and H2B they remain present within the core regions. ठीक है. जो एंट्री है डीएनए की और एग्जिट है इन ए न्यूक्लियस, इट इज थ्रू द H3 एंड H4 हिस्टोन्स. ये एक कॉन्सेप्ट भी आपको याद रखना है, चाहे यहां देख लें चाहे यहां देख लें. ठीक है. नेक्स्ट, देयर इज अ फॉर्मेशन ऑफ we can say structure which is known as 30 nm fiber. It is the next level of organization, we can say, that is the 30 nm fiber. And for 30 nm fiber we are having two different models, that is the solenoid model and the zigzag model. दोनों को समझते हैं, important है बहुत ज़्यादा. क्या differences है? In case of the solenoid model, as we already discussed, there is an arrangement of 6 nucleosome in a helical structure that leads the formation of a solenoid like structure which is hollow from the center. ठीक है, इसलिए इसको अपन कहते हैं it is a solenoid model. Now here there is a helical arrangement of nucleosome. First point, there is a helical arrangement of nucleosome. The packaging ratio is 1:6, means we are having 6 nucleosome in one helix. So the packaging ratio is 1:6. Now here the H1 remain absent in the central axis. जो H1 हिस्टोन है वो सेंट्रल एक्सिस में नहीं रहता, मतलब H1 हिस्टोन आपको कहां देखने को मिलेगा? यू आर हैविंग H1 here, here between the two nucleosome. So it will not remain present in the central axis. जो अपन सेंट्रल एक्सिस की बात करें, टॉक अबाउट द सेंट्रल एक्सिस, जो सेंट्रल एक्सिस है वहां पे आपको नहीं देखने को मिलेगा क्या? यू आर नॉट हैविंग द H1 लिंकर हिस्टोन ओवर द सेंट्रल एक्सिस. तो सेंट्रल एक्सिस के अंदर आपको कहीं पे भी H1 लिंकर हिस्टोन देखने को नहीं मिलेगा. इट्स जनरली रिमेन प्रेजेंट इन द ऑर्गेनिज्म व्हिच हैव शॉर्टर H1 डीएनए. H1 डीएनए अपन ने कहा था कि जो लिंकर डीएनए है वो अपन कह रहे हैं, उसकी बात कर रहे हैं अपन. इट वेरीज फ्रॉम 20 to 60 बेस पेयर. सो द लिंकर डीएनए साइज इट इज फ्रॉम 20 to 60 बेस पेयर. एंड द ऑर्गेनिज्म व्हिच हैव शॉर्टर लिंकर डीएनए, यहां पे अपन ने H1 डीएनए लिख दिया है. ये क्या है? व्हिच इज हैविंग शॉर्टर लिंकर डीएनए. उसके अंदर आपको सोलेनॉइड अरेंजमेंट देखने को मिलता है, वेयर देयर इज एब्सेंस ऑफ H1 हिस्टोन इन द सेंट्रल एक्सिस. ठीक है, क्यों? क्योंकि शॉर्टर डीएनए ऑर्गेनिज्म में क्यों प्रेजेंट होता है? क्योंकि यहां अपन देख सकते हैं कि जो H1 साइट है या H1 की जो साइज है, अगर इस फॉर्म में अरेंज रहना है उसको, तो यहां पे अपन देख सकते हैं कि वी आर हैविंग ए शॉर्टर लिंकर डीएनए साइज. जो लिंकर डीएनए साइज है वो शॉर्टर है, तभी इस तरीके का अरेंजमेंट आपको देखने को मिल सकता है. अगर आप इस शॉर्टर अरेंजमेंट के साथ में जिगजैग मॉडल पे जाएंगे, इफ यू कंपेयर दिस विद दिस, यहां पे क्या है? देयर इज द प्रेजेंस ऑफ 1 nucleosome, 2 and 3 third nucleosome. It will remain present beneath the 1. There is a 4 which remain present beneath the 2. So in this way it appears like a double standard structure. That's why it is also known as the two start model. इसको अपन two start model कहते हैं. There is a zigzag arrangement of the nucleosome. It is also known as two start model. Here the H1 present in the central axis. आप देखेंगे कि जो H1 हिस्टोन है वो आपको सेंट्रल एक्सिस में देखने को मिलेगा. कैसे? बात करते हैं. We are having H1 here, here, here and here and also here. So the central axis, we can say, if we talk about the central axis, we are having the H1 histone over here. ठीक है? So we can observe the presence of a H1 linker histone in the central axis, and the central axis it is not hollow like occur or like found in case of the solenoid model, we can say. Now this type of model or the arrangement it generally favors in the organism which are having the, we can say, which are having the longer लिंकर डीएनए. जिनके पास में जो लिंकर डीएनए साइज है वो ज्यादा है, उनमें ये स्ट्रक्चर फेवर करता है. कैसे? बिकॉज आप देखेंगे कि यहाँ पे जो अरेंजमेंट है, अगर इस जिगजैग अरेंजमेंट में अरेंज होना है न्यूक्लियोसोम को, तो जो लिंकर डीएनए का साइज है वो ज्यादा होना चाहिए. वहीं अगर अपन यहाँ देखें हेलिकल स्ट्रक्चर में, द साइज ऑफ दिस लिंकर डीएनए इट इज वेरी शॉर्ट. सो द ऑर्गेनिज्म व्हिच हैव लॉन्गर लिंकर डीएनए, the favorable model it is the zigzag model, while the organism which have shorter linker DNA size, the favorable model is the solenoid model. क्योंकि यहाँ पे अपन देख सकते हैं कि एक जो आपका न्यूक्लियोसोम है दूसरे न्यूक्लियोसोम के बहुत पास में प्रेजेंट रहता है, और जो लिंकर डीएनए का साइज है वो छोटा होता है. सो वी कैन से दिस इज द कंपैरिजन ऑफ टू टाइप्स ऑफ 30 nm फाइबर व्हिच आर सोलेनॉइड एंड द जिगजैग मॉडल. नेक्स्ट अपन बात करें 300 nm लूप्स के बारे में. सो देयर इज ए इंटरेक्शन ऑफ 30 nm fiber with some non-histone protein. Now these non-histone proteins, these are generally acidic in nature. जैसा कि अपन ने पढ़ा, histones they are basic in nature that are made up of lysine and arginine amino acids. वहीं पे जो non-histone proteins है they are acidic in nature, which include some scaffold proteins. SMCs means structural maintenance of chromosome protein that includes the cohesins and the condensins, जिसके बारे में अपन ने पहले भी बात करी, अब डिटेल में बात करते हैं. So we are having cohesin and the condensin, and the condensin further divided into two types, that are condensin 1 and the condensin 2. Now during the formation of 300 nm fiber, there is a association of we can say there is in further condensation of these solenoids or the zigzag fibers in the formation of loops, and the protein which plays an important role in the formation of these loops, these are called as the condensins, है ना, उनको अपन कहते हैं, these proteins they are called as condensins. Firstly, there is a role of condensin 2, okay, which having the ring type of structure that helps in the arrangement of these loops. That's why these protein they belongs to the category of structural maintenance of chromosome. Chromosome का structure maintain करते हैं. Now the 30 nm fiber, it interacts with these scaffold, that is the condensins, through the regions which are known as SAR regions, and these SAR regions, these are the AT rich regions, और जो outer side में regions होते हैं, these are GC rich regions. So जिन regions से ये attach होते हैं through the scaffold, these are the AT rich regions which are known as SAR regions, that is scaffold associated regions, जिन्हें अपन कहते हैं scaffold associated regions. So due to the, we can say, further condensation, there is a formation of structure that is called as 300 nm fiber, where the 30 nm fiber interacts with some non-histone proteins like the condensins. Now, likewise, in next step, the condensin, they further carry out the condensation of this 300 nm fiber into a 700 nm fiber. After the functioning of condensin 2, there is a functioning of condensin 1 which further carry out the condensation, and due to this further condensation, we can say now we are having the structure which is of 700 nm in size, known as the chromatid. Now this chromatid, it is having a region which is known as centromere. Now the centromere of two chromatids through with the help of, we can say, with the help of a protein which is known as the cohesins. The centromere of two chromatids with the help of a SMC which is known as cohesin, they interact with each other, lead to the formation of a structure which is 1400 nm in size, known as the chromosome. So in this way, there is a formation of a chromosome from chromatin. There is a role of histone and some non-histone proteins. Next, if we talk about some histone variants which are not the core histones, जिसके बारे में अपन ने बात की, that is H2A, H2B, H3, H4, which are forming the nucleosomal core, and there is a H1 linker histone. इनके अलावा भी कुछ histone होते हैं जो variants हैं इन्हीं histones के. तो we are having some histone variants. First is the H2AX. It is the variant of histone, that is the H2A. It generally senses the double-stranded break in the DNA. So we can say these are the sensor for DNA double-stranded break. Whenever there is a double-stranded break in the DNA, we are having the protein which are known as ATM. These are the ataxia telangiectasia mutated protein, which जिसके बारे में अपन बात करेंगे जब अपन cell cycle के बारे में पढ़ेंगे. So we are having the ATM protein. These are generally the kinases which carry out the phosphorylation onto the serine 139 residue of this H2AX. H2AX के serine अमाइनो एसिड के ऊपर जो 139 पोजीशन पे होता है उसको फॉस्फोराइलेट कर देता है कौन? This ATM. And after phosphorylation, this H2AX is now known as the gamma H2AX. Then this gamma H2AX, it binds to the region where there is a double-stranded break. Then it recruits the repair mechanism, and after the completion of repair, they will leave the strand. So in this way, we can say these H2AX, that are the variants of this H2A histone, they help in the process of DNA repair. Similarly, if we talk about the H3.3, it is the variant of H3 histone. We can say it is replication independent. It is having replication independent deposition. It generally attaches to the DNA at the time of transcription. We can say it maintains the transcriptional memory ट्रांसक्रिप्शनल साइट्स को याद रखता है और ट्रांसक्रिप्शनल मेमोरी बनाए रखता है, मतलब कहाँ कौन सी एक्टिव साइट्स हैं जहां पे वापस ट्रांसक्रिप्शन अपने को करना, मतलब किसी जीन के एक्सप्रेशन की आपको वापस जरूरत पड़ गई तो उसको याद रखने के, मेमोराइज करने के लिए वी आर हैविंग द H3.3. Next, it facilitates the repair and reactivation of transcription after the repair. So we can say if we are having the double-stranded DNA, then there is a transcription, and during the transcription, there is a binding of RNA polymerase and synthesization of mRNA. Now, during this, there is a recruitment of, or we can say there is a replace of this H3 by H3.3, and this H3.3, it helps in the recognition, or we can say it helps in the memorization of the active sites, as well as if there is a damage during the transcription process, it stops the transcription, it recruit the repair mechanism, and after repair it reactivate the transcription. तो ये भी एक तरह से अपन कह सकते हैं इन्वॉल्व होता है रिपेयर में during the process of transcription. Now the third one, it is known as CENP1, that is centromere protein 1. Again, it is the variant of H3 histone. During the interphase, we are having the interphase chromatin, which will further condense into the metaphasic chromatin, that is known as the chromosomes. The two chromatids, they remain attached with each other through their centromeric region with the help of SMC, which is known as cohesin. Okay? Now, this CENPA1, it recognizes the centromeric regions. After recognition, it recruits the CENPA, and the CENPA then interacts with the kinetochore, and these kinetochores, they further interacts with the microtubules, which carries out the movement of these chromatid after the breaking of this cohesin into the different poles during the cell division. Cell division में अपन कहते हैं काइनेटोकोर के ऊपर अटैच होता है माइक्रोट्यूब्यूल, तो काइनेटोकोर को सेंट्रोमियर से अटैच करने के लिए जो प्रोटीन हेल्प करता है, दैट इज the CENP1 protein, which is centromere protein 1, which is the variant of histone H3. So these are the variants of histone. Now the last thing is that we are having two type of chromatins. Now one is known as the euchromatin, while other is known as the heterochromatin. If you carry out the staining of the chromatin, the regions which appears dark, these are known as the heterochromatin. These are the dark stained regions, while the regions which appear light, these are called the euchromatin regions. Euchromatin regions are the regions where the DNA, it loosely attaches to the nucleosome or the histone, and the heterochromatins are the regions where the DNA tightly attaches to the nucleosome, that is a condensed chromatin we can say, while the euchromatin, it is a loosed chromatin. Now it is loosed, so it is early replicating, पहले replication होगा, while these regions which are having the heterochromatin, these are late replicating. Similarly, these are transcriptionally active, means there is an expression of gene, while these are transcriptionally inactive. So we can say it is one of the way to carry out the gene regulation. अगर किसी जीन को एक्सप्रेस कराना है तो उसको यूक्रोमेटिन में कन्वर्ट कर देना चाहिए, अगर किसी जीन को सप्रेस करना है we can convert it into the heterochromatin. स्ट्रक्चरल डिफरेंस देखते हैं, यहां पे अपन देख सकते हैं there is a loose binding of the DNA with the nucleosome, while there is a tight, देख सकते हैं यहाँ पे there is a tight binding of this DNA with the nucleosome in case of heterochromatin. So if you want to convert a heterochromatin into the euchromatin or a euchromatin into the heterochromatin, for this we are having two processes. One is called as DNA acetylation, while other is called as histone acetylation, while other is called as DNA methylation. Simply, अगर आप हिस्टोन को एसिटाइलेट करेंगे, मीन्स इफ यू ऐड this acetyl group onto the histone, and the acetylation it generally occurs on the lysine amino acid, और जो lysine amino acid की side chain है which are having the NH3 group, अगर वहां पे आप acetyl group ऐड करेंगे, you are reducing the positive charge of the histone, और अगर आप पॉजिटिव चार्ज को कम कर रहे हैं, इसका मतलब जो बाइंडिंग है डीएनए की और हिस्टोन की वो लूज हो रही है, ठीक है। सो दे जनरली न्यूट्रलाइजेस द पॉजिटिव चार्ज ऑन द लाइसिन अमाइनो एसिड। लाइसिन अमाइनो एसिड के पॉजिटिव चार्ज कम कर देते हैं। लाइसिन अमाइनो एसिड पे ही जनरली एसिटाइलेशन होता है। किस हिस्टोन पे होता है? H4 हिस्टोन के लाइसिन 8 पर और H1 हिस्टोन के लाइसिन 16 पर। These are the major acetylation sites, and the acetylation of histone, it reduces the positive charge and thus it will convert a heterochromatin DNA into the euchromatin. Similarly, if we talk about the DNA methylation, if you add the methyl group onto the DNA, then it will again we can say neutralizes their negative charge. वो क्या करेगा? न्यूट्रलाइज करेगा नेगेटिव चार्ज को एंड इट विल इंक्रीज द पॉजिटिव चार्ज। द मिथाइल ग्रुप इट विल इंक्रीज द पॉजिटिव चार्ज ऑन टू द डीएनए। पॉजिटिव चार्ज बढ़ाएगा तो नेगेटिव चार्ज को कम करेगा। अगेन जो इंटरेक्शन डीएनए और हिस्टोन का होगा वो लूज होएगा एंड देयर इज अ कन्वर्जन ऑफ हेटेरोक्रोमेटिन इनटू द यूक्रोमेटिन। नाउ द मिथाइलेशन इट जनरली अकर्स ऑन द साइटोसिन बेस। जो एटीजीसी अपन ने बात की थी, जो साइटोसिन है उसके ऊपर मिथाइलेशन होता है and for this we are having a methionine, modified methionine which is called as S-adenosine methionine, जिसके बारे में अपन ने अमीनो एसिड्स में पढ़ा था। The methyl group it is taken from the S-adenosine methionine which carries out the methylation of DNA through an enzyme which is known as DNA methyl transferase. So we are having two conditions. If you want to convert a heterochromatin into the euchromatin, तो क्या करना चाहिए अपने को? We have to carry out the acetylation of histone. हिस्टोन का एसिटाइलेशन कराना है, कौन करेगा? हिस्टोन एसिटाइल ट्रांसफरेज करेंगे। दे विल ऐड द एसिटाइल ग्रुप ऑन टू द H4 हिस्टोन लाइसिन 8 और H1 हिस्टोन लाइसिन 16, और जैसे ही वो ऐड करेंगे पॉजिटिव चार्ज रिड्यूस हो जाएगा एंड देयर इज ए लूज बाइंडिंग दैट विल फॉर्म द यूक्रोमेटिन DNA. Similarly, if you carry out the DNA methylation with the help of enzyme DNA methyl transferase, if you add the methyl group onto the DNA, it again neutralizes the negative charge and will convert the heterochromatin into the euchromatin. अगर vice versa करना है, means if you want to convert a euchromatin into the heterochromatin, तो क्या करना पड़ेगा? हिस्टोन पे से एसिटाइल ग्रुप को हटाना पड़ेगा। So for this we are having the enzyme which is known as histone deacetylase that removes the acetyl group from the histone, that increases the positive charge, and there is a tight interaction between the DNA and the histone and will convert into the heterochromatic form. The another enzyme which carries out the reaction in this direction, it is a DNA demethylase which removes the methyl group from the DNA. Similarly, the removal of methyl group from the DNA, it increases the negative charge on the DNA, and again there is a tight binding of nucleosome and the DNA, and there is a conversion of DNA into the heterochromatic form. So this is all about the genome organization in case of eukaryotes. Thank you very much.